So thanks for coming. This is a good turnout. So we have a great talk tonight. Speaker Lindsey from Amy Keating's group. Lindsey's been in Amy's group for a couple of years now. She's working towards her dissertation. And this is a project she was talking about that's along the way of her fully computational work in her PhD. Before she was on the East Coast, she spent 4 years at UC Berkeley on the West Coast. Probably really, being that it's April 9th today and was freezing this morning, you might be missing California a little bit. Thanks, Lindsey, for giving this talk. So Lindsey's going to talk to us about her, tinkering around with AlphaFold 2 and what it understands about proteins and peptides. So with that, looking forward to talking.
Thanks. Thanks, Nick, for that introduction. Thank you so much to the organizers. I really like this talk series, so it's a great honor to be able to give a presentation. So today I'll be telling you about, some things that we've discovered about these state-of-the-art structure predictors and how they work for protein-peptide complexes. And I'm sure this is not an unfamiliar concept to most of you, but protein-peptide interactions are really prevalent across different biological systems. And here's a sampling of some of the things that we study in the Keating lab. There's a huge challenge in predicting protein-peptide structures, kind of historically. So for protein-protein docking, sometimes we can make this assumption that these chains will fold individually, and then we can do something kind of like rigid body docking to get a docked complex structure. Of course, that's an assumption, but an assumption that we can sometimes make. But with flexible protein peptide docking, that peptide can adopt so many different conformations that may be difficult to understand unless you have solved a co-crystal structure or something like that. And so just the conformational space can be much greater if you don't know what this peptide is supposed to look like bound. But AlphaFold 2 was, of course, a huge breakthrough for the protein folding community. And at first, people realized that, oh, you could just connect two chains with this glycine hack, and then it will be able to fold complexes for you. But then DeepMind came out with this multimer model so that you don't have to do this hack, and it was trained on multimeric structures, and then it became the state-of-the-art in structure prediction. And this was really the tip of the iceberg, because now these structure prediction models have been used to do not just structure prediction, but also design. So AlphaFold 2 has been used as a scorer to rank different designs and has been inverted to do binder design as with Minecraft. But we still consider this a pretty unique challenge, even with all of these different computational tools at hand. And this is because these models still rely on an MSA. And when we consider protein peptide complexes, the peptide often has a sparse MSA. And with this newer generation of models, even though there's a lighter weight module that processes the MSA, people have seen that they definitely still rely on evolutionary information, often in the MSA. And so, as I mentioned, this peptide MSA is often sparser, and this is due to mostly two reasons. One, sometimes we're defining peptides as short sequences, and so you just get fewer statistically significant homologs. And second, peptides often come from intrinsically disordered regions, and those are subject to different evolutionary constraints and can be difficult to just make MSAs for. And so there's less evolutionary information for peptides. This makes it kind of out of distribution for what these models are used to seeing. So how does it work, and does it actually work for protein peptide complexes? And if they do work, what information do these models rely on? And these are the questions that I'll try to answer today. So first, how good are these models really? First, I curated a data set of solved protein peptide structures. So just querying the PDB with a bunch of different filters, considering things like cofactors or ligands that might be interfering with this binding site, or crystal contacts that might be influencing the way that the peptide is binding. And then we end up with about 600 protein peptide complexes. And to assess how well a model is doing for this task, I'm going to be using this metric .q, which has been used in the docking community or protein docking community as a measure of accuracy. And it incorporates three things: a ligand RMSD once you align the targets, interface RMSD, and also the fraction of native contacts recovered. And they're combined in this way. And so now I'm going to show you a bunch of examples to kind of calibrate our sense of what DocQ is. First is one where the prediction is correct and it is very confident. And so this prediction is from AlphaFold2 multimer. So DocQ 0.95, that's basically like a perfect prediction, and confidence of 0.97, that's also very highly confident. Second is this example where the binding site is correct, but the peptide pose is not quite there. And we see that reflected in the DocQ in 0.5 and a confidence of about 0.6. And this prediction came from Chai. This is one where it's a very incorrect prediction, and both the DocQ and confidence reflect that, and the failure mode here is that the target was not predicted correctly. And finally, this is an example where actually the prediction was very incorrect, but the model was pretty confident about it. So the binding site is totally incorrect in addition to the pose. And these are what we consider like the most concerning examples.
For these kinds of computations you're doing, were you also having the model predict the structure, or were you using a template for the protein? Or were you having it predict both the protein structure plus the peptide at the same time?
Yeah, we're asking to do both. So no template provided. Yeah. But overall, performance is excellent. We see that the vast majority of predictions are in this range of greater than 0.5 DocQ, and so it's good or almost perfect. And there's a pretty good correlation with confidence. And so that's great to see, very promising for models across the board. But there were still some of these predictions that were concerning. And this one sort of raises the question of, okay, this peptide, not only is the binding site wrong, but there's also this helix that we don't expect it to form. And that led to this question of, do models tend to make up helices or just make up secondary structure that doesn't exist? And to do that, we took the peptide in the native and predicted, or in the native structure and in the predicted structure, calculated what % secondary structure composition it had, looked at both native and predicted, and we can make this kind of plot. So here I'm showing the fraction of peptide for each structure and how much of it was helical. And I'm doing this across the models, and we see that most peptides, or for most models, peptides are predicted to be more helical than they actually are. And we can look at this for sheets, and for most models that actually there's not a significant difference, but AlphaFold3 does predict residues or peptide residues to be more sheet-like than they actually are. And all models predict fewer residues to be loops than they actually are. And so, yeah, models predict peptides to be more helical than they are. Models predict peptides to be less loop-like than they are. And so this was just one kind of symptom that we noticed from looking at these predictions. We also wanted to know if there was a correlation between what secondary structure to expect and how accurate these models were going to be. And for that,
Why they do this?
no, not quite. I think there's a relationship between just like how stable it thinks a structure is and how likely it is to make a helix. Like a helix can be stabilized kind of locally, and so it thinks that if a peptide can do that, then it likes that structure. But I don't know if I can say, exactly why it does that. Yeah. So for this experiment, we looked at interfaces that were either mostly helical, mostly sheet, or mostly loop, and looked at how well these models could predict the structures. And all models just generally predict loopy interfaces more poorly than, for example, helical interfaces. And so we can say that interfaces with irregular secondary structure are predicted less accurately. And so this is another example that was a little bit concerning to us. And for For a different reason. So this was a perfect prediction. The target here is a urokinase, and urokinases have been a drug target, and there are a lot of solved structures in the PDB of urokinases and cyclic peptide inhibitors. And although this specific structure, like PDB 6A8N, although that was released after the training date, it's still predicted perfectly, and there are a lot of similar structures in the PDB. And so that leads me to our next question, which is how much of the model success that we see can be attributed to memorization of the training set? So kind of a classic question, is a model just memorizing? And to do this, we took every test structure that we had. We searched against the PDB pre the training day cutoff used in each model and found the training set matches, and we were able to count them. And so when we look at the performance of each model split by how many matches we saw, so either 0 matches or greater than 0 matches in the training set, we see a significant difference in that completely novel interfaces were predicted worse by each model. And conversely, highly represented interfaces are predicted more accurately. Yeah.
Do you have a sense for the raw counts, like how many models have 0 matches?
Yes. So it's different for AlphaFold 2 because they use a different train cutoff, but it's about 5, I want to say like 590 and 40 for all of these, like give or take some depending on the train date. I don't have the exact numbers. Yeah. So actually like very few have 0 matches comparatively. Okay, so this is maybe not that surprising, like this result, because every machine learning model probably has these kinds of biases toward the training set, but this kind of issue can manifest in a different way, and so that leads me to the next question, which is, can training set matches actually mislead predictions? Like, can we tie this in to the incorrect predictions that we see from each model? And so what we can do is we can look at all the wrong predictions and count the matches if we search for the structure corresponding to those wrong predictions. And so in yellow, I'm showing the fraction of wrong predictions where actually the prediction is better represented in the PDB, and in blue, those are the structures where the native was better represented than the PDB. And so for each model, it's about from 10 to 30% where actually we might be able to say that the training set sort of misled the prediction. And I kind of want to show some examples because these are fun to look at. So first example, this is a structure where the target is expected to bind both a ligand and a peptide. If we provide bolts with just the target and the peptide, then the peptide goes into the site where the ligand was supposed to go. That site is actually more represented in the PDB as a peptide binding site than where it actually goes in the native structure. But this is also kind of an unfair test. We didn't give bolts the ligand, and so maybe if we gave it the ligand, it would put the ligand in the right place and then put the peptide in the right place. But that's not really true. If we give bolts the ligand and the peptide, it puts the ligand in roughly the right place, but then the peptide somewhere else, but still incorrect. And so here, there's still, yeah.
What now are they from data that is outside of the training cutoff?
Yes, here it is. Yeah. Like post the like 2021 or yeah.
So just from my based on the angle of this, it almost looks like a green native peptide. Part of it might be out into space and not interacting necessarily with the protein. Maybe it's part of a crystal contact or something in the crystal. So I'm just curious is how much of that peptide is actually engaging with the gray protein?
Yeah, that's a good question. So as part of my filters in creating this set, I do enforce like a surface area, like interaction area cutoff, and I also limit the number of crystal contacts that the peptide engages with. So it's possible there's something interfering with it, but I did try to check for those things specifically. Yeah, that's what I can say, but I don't know about specifically like this complex, like what the contacts are like. Okay, so second example, this is the structure of an important alpha and a peptide that it binds. The thing about these important proteins is that they actually have two binding sites, a minor site and a major site. And specifically for this peptide up here, the folks who deposited the structure, they found that this is specifically binding to the minor site and doesn't bind the major site. But both the minor site and major site are represented well in the PDB. And what does AlphaFold 3 do? It puts the peptide in the major site. So this is the same peptide as in this top structure here. It puts it in the major site and then wraps the N-terminus of the target back into the minor site. So it really likes to try to fill the binding sites that it thinks exists on this target, which is kind of an interesting pathology. So, okay, what do we know so far about how good are these models really? Overall performance is great. These are absolutely the state-of-the-art models, but there are a few potential pitfalls. So if there's irregular secondary structure or this is a novel interaction, it could be harder for these models to do well. And if you don't have a novel target, there are cases we see where it gets stuck in a binding site it has seen before. Okay, so that's question one and our understanding of how good these models are, really. So the second question, what information do these models rely on? So of course, these things that I've observed here, they don't explain every single prediction. And of course, we still see like these totally novel interfaces being predicted accurately. So what's going on and what information makes these models work well? And for this, I'm really going to focus on the MSA. So the MSA, it's broken up into these two parts that I'll refer to. The first is a paired region, and the second is an unpaired region. So for the paired region, the sequences between two chains that correspond to each MSA, they're paired up by species. And in the unpaired, they're on the block diagonal. And so every other sequence that couldn't be paired by species gets thrown in there. And first, I want to think about the paired MSA. And so the reason that these models bother to do this species pairing is the idea that interchain coevolution might inform predictions. So the same mechanism that we think might be driving predictions within a chain could also be driving predictions between, or contact predictions between chains. But in our set, only 2 complexes had a paired MSA with greater than or equal to 50 sequences, and only maybe like 12 that had greater than 0 sequences at all. But this doesn't really answer the question of whether the pairing would be useful if we had access to deeper paired MSAs. And so how do we make deeper paired MSAs for peptides? The way that people have done this, particularly in the slim domain interaction community, is they've been able to add sequence context to the peptide. And so in the PDB, the structure will have some short region, but that region will be extracted from a longer protein that may evolve together with the target in the same species, for example. And so if we take the delimited peptide from the PDB, add 100 residues of context around the peptide, and then use that extended sequence to build the paired MSAs, then we can get deeper paired MSAs. So we could do that, extract the peptide residues, and then make these predictions again, now with a deeper paired MSA. And we can now ablate this new pairing by shuffling the MSA so that it's no longer paired by species, and that should obscure any coevolutionary information that came from this pairing procedure. So we find that yes, if you shuffle the paired MSA, covariational signal is reduced. And so here I'm reporting mutual information between the chains, so it just interchain mutual information. But we see that doing the shuffling thing does not significantly reduce performance. And that seemed to be the case for basically every model. And so that told us that maybe it doesn't really rely on covariation. But we had to do this kind of hacky thing to make these deeper paired MSAs. We could actually just look at the protein-protein complexes that already have good MSAs and do the same thing. And so that's what we did, just to kind of validate this result. So again, we see that shuffling. So we're looking at this set of like 340 structurally solved protein-protein interactions. These folks in this paper have already compiled this data set. And so this shuffling procedure does reduce covariation, but it doesn't make a huge impact on performance when we shuffle. There's a bit of an impact for AlphaFold2 and bolts, but for AlphaFold3 and chai, it's not a significant difference. And so shuffling the paired MSA only slightly reduces performances on protein-protein interactions, which is perhaps surprising because so much of what people thought went into a good protein-protein interaction prediction came from being able to draw conclusions from paired species or pairing by species.
Is the performance the same if you have really shallow or no MSA for the peptide?
I think I will get to that.
Oh, you will?
Yeah.
Seems like peptide.
Yeah. I think I'll get to that. Yeah. Yes. Here I set a cutoff of like 50 residues. Yeah. Yes.
I may have missed this, but here is your test set filtered by protein and peptide from the same species.
It's not. Yeah. So it's purely by like length, and yeah, no species filter was done. So some peptides are like synthetic. Yeah.
So I wonder if you see more correlation or difference here.
Well, specifically here, it's with protein-protein interactions. Oh, are you asking about this set?
Yeah, for like the species coevolution.
Yeah.
Protein peptide pairs.
Yeah. I don't do any species filtering. Mhm. I feel like I saw another hand. Okay. Yes. Good question. I haven't tried other MSA methods. This is like the MMSeeks from Collab Fold. Yeah. Okay, so the first thing we've learned about what information these models rely on, well, covariation from species pairing across chains doesn't explain successes. Paired MSAs are so sparse. Anyway, sorry, I don't know why this got cut off. But even when that pairing does exist, it doesn't seem to matter very much. And we've seen that with both protein-peptide complexes and with protein-protein complexes. But I'm not actually the first person to make this observation. I think that at CASP 15, there were a lot of methods that didn't rely on species pairing, and they found that there wasn't a huge difference in performance for them either. So this is sort of an emerging theme in the field. Okay, so that's all for the paired MSA. Now, moving on to this unpaired MSA, and more specifically the target MSA, and this might be the most relevant question if you're interested in designing binders to targets, because these the targets that at least we're interested in are ones that are naturally occurring or evolved. So we see that the target MSA is necessary for decent performance. This also isn't surprising because without the target MSA, often the target can't be folded. But is the target MSA also sufficient? And this is kind of getting at Nick's question of like, can it make a docking prediction with just the target and no information about the peptide? And so to do this, we kind of took it to the extreme where we remove all information except the target MSA, and that includes the peptide sequence. So we can mask the peptide sequence with the mask token X. And from AlphaFold2 multimer, we can get contact maps. So we can't actually get the backbone structure out of the box with AlphaFold, but you can get this histogram. And what you can do is you can look at the interchain contacts and compare those to the native contact map. And I came up with this metric to compare these contact maps, and somewhat arbitrarily, I've decided that if the RMSD map value is less than 3, then it's a pretty good prediction. It does correlate with .q for complexes or for structure predictions where we do give it the sequence. And so for now, you'll just have to take my word for it that less than 3 is pretty good. For bolts and chai, you can actually get the backbone structure when you mask the residues. And so we can compare those instead of having to go through a contact map. and look at a peptide RMSD. And here I consider less than 2 a good prediction. And here's some examples to kind of calibrate your sense of what these scores mean. So here in the top here is an interchain contact map where the dot Q was very good and we see a low RMSD map with the sequence provided. If we then mask the sequence, this is what the contact map looks like, and this is what the native looks like. For this example with chai, we see that there is a pretty good dot Q with the sequence provided, a low RMSD, and also a pretty low RMSD, although you can see what the issue is here when you mask the sequence. It has flipped the peptide in the binding site, but the ligand RMSD or the peptide RMSD calculation doesn't consider that. It just aligned the backbone atoms anyway. And finally, for this example with bolts, we see that the masked peptide resulted in just like a totally wrong prediction with maybe very slight overlap in the binding site. And so that produces a pretty high peptide RMSD. Okay, so what do these predictions look like? On the x-axis, I'm showing what this metric would look like if you provided the peptide sequence. And on the y-axis, I'm showing what it would look like if you masked the peptide sequence. And we see that for everything to the right of this dashed line, which is everything that was pretty good with the peptide sequence. There are about 50% of predictions that remain good after you mask the peptide. And we see something kind of similar with bolts and chai, where things that were good, about 50 to 60% were still good after masking. And tying this back into the idea that maybe sites are being memorized or there's some amount of memorization going on, are common binding sites being predicted better in this masked peptide regime? And so doing the same test as before, like counting training set matches, we see that for AlphaFold multimer, there doesn't seem to be a huge difference between in the training set matches between unsuccessful and successful mask predictions, but with bolts and chai, there does seem to be a difference in that structures that were unsuccessful after masking had fewer training set matches. So yeah, there's some amount of memorization going on, but we still see examples like this where with the sequence provided, the prediction is good. With the sequence masked, the binding site is still identified, but there were no similar interactions in the training set. So just purely based on the target MSA, AlphaFold was able to return a binding site prediction, basically. And so what is it about the target MSA? Like, what information does that have that can inform these kinds of decisions? And to answer that, we made the question a little bit smaller in scope, which is docking based only on target structure also evolutionary information? And here's what I mean by that. If we take a look at predictions made with just the target MSA in the masked peptide, that's where we provide evolutionary information, but the model has to infer structural information. If we give it the template and no MSA and the masked peptide, it has no evolutionary information, but it has perfect structural information. And so if we compare performance between these two conditions, we can see whether or not it really needs that evolutionary information in the MSA, or if it can just do all of this by getting a good understanding of the target structure. And we see that performance does get worse when you just provide the target template and you don't provide the target MSA, showing that there's something more in the target MSA than just the way that it informs the structure prediction of the target. And we can bring it back to the more realistic scenario where you don't mask the peptide, you actually just give it the peptide sequence. And for this, we can compare both AlphaFold 2 and AlphaFold 3. And we see that, yeah, for AlphaFold 2, predictions are much worse when you only give it the template, although this difference isn't significant for AlphaFold 3. And we do know that the targets are being predicted better when you give it the target template. And so what is that evolutionary information that the model seems to rely on? So our binding sites chosen based on conserved residues on the target. And we ask this because residue conservation has been used in a lot of different models to predict binding sites on targets, and it is sort of an obvious choice for trying to understand what statistics could come from just one MSA. And we see this example with AlphaFold 2, where actually the prediction does seem to be in a more conserved region. So in green here are the native binding sites, and in gray are the conservation of the residues that it's interacting with, and in blue are the predicted. And so the prediction is being placed in a more conserved region. But for every example we have like this, we have an example like this, where actually the prediction is not in a more conserved region. And overall, there isn't much of a difference between predicted binding sites and native binding sites in terms of how conserved the sites they chose, or in terms of how conserved those residues in those sites are. There's a bit of a difference for AlphaFold 2. So taking inventory of what we know from the target MSA. So the target MSA has a ton of information, and that's sometimes sufficient to predict or memorize binding sites. And for AlphaFold 2, providing the target template isn't as important as providing the target MSA, indicating there's some further evolutionary information that makes these predictions better. And that evolutionary information, well, it's not necessarily conservation alone, because that doesn't explain the way that these models choose binding sites. Okay, now briefly about the peptide MSA. So we're able to make a peptide MSA for about 150 of the structures in our set. And for these, when we ablate the peptide MSA, we see that across all the models, performance does get a little worse. So we do know that the peptide MSA does still contribute to good performance. And to try to answer that, we asked, or try to understand why this is true, we asked, does the peptide MSA still matter if the binding site is known and provided? So does it help predict the binding pose of the peptide, or is it useless if you already know the binding site? And to answer that, we use this handy feature from bolts and chai. So with bolts, you can actually specify some pocket residues on the target, and it will try to put the peptide or try to put whatever other binding partner you have in that pocket. And with chai, you can specify distance constraints. So with bolts, I'm going to provide 3 pocket residues, and with chai, I'm going to specify 1 restraint. And we see that even with these binding site definitions, even though we've told it exactly where the binding site residues are, the peptide MSA still helps. So when we ablate it under these conditions, it still drops performance for a good number of these structures. So yes, the peptide MSA can still inform a docked pose prediction. So kind of stepping back and looking at this unpaired MSA, which is still useful if you're able to make an MSA for your peptide, even if mostly you care about a situation where you're like designing a peptide, you might be interested in whether or not these 2 proteins from different species interact, which is why we in the Keening lab still care about that. Well, so how is it combining information from the protein MSA and the peptide MSA? And this is sort of an open-ended question, and there are a lot of ways you could probably answer this question, like trying to understand exactly what architectural decisions were made in these models. But also to limit the scope of this question, we asked, well, without a paired MSA, can the MSA modules still do interchain reasoning? So we know that the target MSA already has a lot of information, and we know that the peptide MSA can improve predictions, but is that happening through the MSA module, or are we somehow just giving the model more information that it then uses in the structure module or diffusion module? And so for this, I pose 2 hypotheses. The first is that, so after the evoformer, and so we're going to focus on AlphaFold 2 for this. After the evoformer, does the model just have a better understanding of these chains alone, and then that information gets passed to the structure module and then gets used? Or is it that after the evoformer, it took those two unpaired MSAs and used those to inform the representation of the chains together, getting some kind of co-folded understanding of the input. And then that is passed to the structure module and then refined. So these are the two hypotheses. And to test this, we used this handy feature that you can do, which is you can modify the attention matrix. And so each value in this attention matrix represents how much each position in your input attends on another position in your input. And you can add this bias to basically cancel out all of the interchain attention in this matrix. And so by doing that, we can basically remove the ability to do interchain reasoning, but allow the model to still get an understanding of what each of these chains look like individually. So when we ablate interchain attention, we do see that performance drops for AlphaFold 2. And that, to me, supports hypothesis 2, where the unpaired MSA actually still enables the model to develop a good understanding of these chains together, even though there's no explicit covariation as defined by what you could get from species pairing. So that was pretty cool to us. And I want to conclude with, I guess, a few remarks about the MSA. And so we know that the models are subject to some bias based on the training set, but of course, this is often true for every machine learning model. And even so, some fully novel predictions are accurate, and that led us to wonder where that information might be coming from. We know that MSAs boost docking beyond just improving the structure of individual chains. And there's kind of a non-obvious mechanism for this, since we were able to rule out conservation and mostly rule out covariation from pairing. And what does this mean if you're a peptide binder designer? Well, maybe we should all be checking for other common binding sites, because that could be informing the way that our models decide where to put the peptide we've designed. And if at least one of your chains is evolved, then MSAs can still be valuable. If you're using these deep learning methods as a scoring metric, then getting better predictions from an MSA may be improving the way that your models are acting as scorers. So with that, I'd like to thank Amy, my amazing advisor, and everyone in the computational subgroup in the labs with Foster, Dustin, Jackson, and Colin, my committee, and my funding sources. Thanks.
So this was an awesome talk, very thought-provoking for me at least. One question I have is sort of just a speculative question, which is, do you think that adding the MSA information just might make the model, the input data, more in distribution for how the model was trained, so that then it performs better because the input is more in distribution? It's seeing more of MSAs in training.
Yeah.
I don't know what fraction of training examples it doesn't see an MSA at all. But I wonder, like, if you were to retrain bolts or something without using MSAs and only using templates, if you would see this performance drop compared to training. So I'm just curious if, like, have you thought about just that the input data is more in distribution of what the model's expecting to see?
Yeah.
That might increase the performance of the model.
Yeah, I think that's entirely possible. I think, as you mentioned, like retraining would be probably the surest way of understanding that. I guess it's hard to know like how you would quantify like what the model thinks is in distribution. Yeah, that's a good point.
What I mentioned is like has implications for protein design, because we usually use single sequence modeling
Right.
protein design, but should we be making pseudo MSAs to feed into the model if it's gonna perform better.
Yeah.
Because it's used to seeing it. I'm just, it's sort of a, at this
Yeah.
point, very speculative. But
I guess that's like one way you could go, like trying to come up with pseudo MSAs, but I guess ideally we could be moving our models toward less MSA dependent architectures, but yeah, yes. Yeah, that's also one option. Yes, I'm so sorry. I've totally forgotten. Yeah, the question was like whether we should just be using language model embeddings in situations where we can't find an MSA. And yeah, I think that's totally one option. I've done like a little bit of preliminary comparisons between like chai with ESM embeddings versus chai with an MSA, but like no peptide MSA. And the difference isn't huge. Like they perform sort of on par. So then you end up with the question like how meaningful is a language model embedding for like a really short sequence? And it might end up being like a very similar question that you end up with. Yeah.
Go back to that slide that showed the data showing when you did the ablations at interchain attention.
Yeah.
I was just wondering if you could comment on like the very large variance you're seeing in your data, sort of like those grouping outliers. Maybe you can talk much about what those are, what's going on in those cases.
Oh yeah. So these would just be predictions that were like bad by default, and then they were still bad after the ablation. Yeah. So I guess those aren't super surprising. Like if it couldn't make that prediction with interchain attention, then it probably wouldn't be able to without. Yeah. Yes.
If I understood correctly, you're saying MSA is not very important for these interactions. Is there an advantage to, in terms of cost to leave it out of these programs, and how much you gain if you ignore it?
Yeah, so the question was, how much should we gain in terms of like computational cost if we just ignore the MSA? I'm sure that, yeah, you would save a lot of computational or like save a lot of time. But if we completely ignore at least if we completely ignore the target MSAs, then performance is just awful. Mostly because it can't fold the target. And in terms of the peptide, I guess since they're so short, you don't save that much time by trying to omit the MSA in terms of like how long it takes to make that prediction. And we do see that it helps to provide it if you have it. So I guess my recommendation would be like, if you have one, then you should use it.
Ask a review question, but have you ever tried looking, you have a lot of ablation studies or mask studies, have you ever tried a neutral test instead? If you're trying to look for an active site right there, why not try a random residue that's not the active site right there and see if you have an ablation that's close enough to that. If I'm right in front of me there, that you see the peptide put into another section that for whatever reason is energetically part of the landscape, but it's not the active site that you were looking at right there. So what is more has to be useful to compare some of you ever tried?
Yeah, so let me see if I understand the question. So are you suggesting like a more neutral test in terms of like this mask token is a little too special? Or
if I just don't have the peptide in there, or have mask right there, or if I have no knowledge of this active site where I'm trying to target and looking at right there.
Yeah.
Would the model place the peptide in any random configuration that is energetically stable?
Oh, I see. Okay. So the question is like, if there was no previously known binding site, where does the peptide go?
The simple
Yeah.
model where the active site will be somewhere completely random.
Yeah, I don't think these models choose somewhere completely random because we do still see instances where there was no previously observed interaction with this target and it still got the prediction correct. And so it wasn't, I guess maybe it was random and it got lucky, but I think that's maybe unlikely. And so there is still some kind of informed decision being made. I don't know if that answers the question. Yes.
For antibodies, they've seen that seeds, like seed, like random samples, they've seen trying to over and over again.
Yeah. Yeah, so the question is, people have seen that if you try a lot of different random seeds, you can get better antibody predictions. I'm sure that is the case with these complexes, but since we wanted to do like so many experiments with a set of like 600 structures, that we just kind of made this decision to like not do super sampling. But I'm sure like, yeah, I'm sure it would help improve a lot of these predictions. Yeah.
Antibodies and MSAs large subsample two can provide a lot of variation. The other variation is because of target MSA.
Yeah. Yeah, I haven't played around with that. Like subsampling the peptide MSA or even subsampling the target MSA beyond just like what it does automatically within these models. But yes, I think that would be super interesting and definitely would affect the predictions. Yeah. Yeah.
So one thing I was kind of curious about is like, I guess in training, there's also like domain interactions.
Mhm.
And I was wondering if you sort of looked at cases where it's able to work, is it possible that it's seen that interface maybe in a different context, like more like a domain interface? Might have to probably chunk away parts of the helices or something to just find that interface really. I'm not sure how easy it is to find them.
Yeah, yeah, the question was whether I've tried to look for domain-domain interactions that look like structures in my test set. Yeah, I think that's a really good idea. But the way that I look for these matches is I look for things that are bound to this binding site but exist on a different chain. And so I can't use what I have right now, but I guess something I could do is like I could link the chains and then look for structural matches so that I could just use something like fold-seq to find structural similarity. But yeah, I haven't tried very hard to look for domain-domain matches. Yeah, good idea. Yes.
Here's about side chains. So I don't know, we're using background RSP or full out of RSP. You're comparing native.
For the question was whether I was using backbone RMSD or sidechain RMSD. For .q, I'm using whole or all atom RMSD, but for these mass predictions with bolts and chai, I'm using just backbone RMSD. Yeah.
Yeah, I guess I'm just curious, like, when I use these models to predict complexes, oftentimes, you know, if I get something that's correct, but the side chains are wrong, which is like interesting, maybe kind of spooky. And I'm wondering, like, do you see any relationship between like that specific aspect of the model, like side chain interaction, whether they're correct, and like how good the model is? Or does it seem like it's almost like the performance of the model is independent of specific side chain contexts?
Yeah, that's a good question. The question was whether like it looks like the model is making accurate specific sidechain interactions. Is that the question? I think that would be reflected in the .q. Like .q enforces, well, so the all atom RMSD could play a role, but also the idea that you want to recover the native contacts. And I think the way they define that part of that would enforce that you're making the right sidechain contacts. But I think something interesting in that is also like when it's memorizing, did it memorize every atom or did it memorize the backbone? And so if you're memorizing, are you like plateauing at a certain level of performance? Yeah, I don't know. I don't think I've done very much in looking at like sidechain packing. specifically. Yeah.
Do you think that this could be standardized in a sort of benchmark, leaderboard, and similar to like elements when you want to come out, they pit them against the other and medical exam or whatever. Do you think that this could be turned into something like that? So let's say I'm the most one, and I just try and see how it performs on all the steps.
Yeah, that's a good question. The question was whether like this benchmark could be standardized to be used across models. I actually haven't thought much about that, although I think I did get a question, or Amy got a question similar to that recently. Yeah, I think this data set that I use, like these 600 structures from the PDB, could be reused for this test, and then the subset of them that are like fully novel, have no like fully no similarities in the training set, like that could be useful, but that set is still kind of small. It's like 40 structures that are fully novel. And so I don't know how much people want to benchmark against a set of like 40 structures, but I guess with the 600 set, like, yeah, that could, if that's useful for people, yeah. Yes.
So, I'm still very intrigued by this finding that you don't need co-variation between the two chains. And you showed it for peptides as well as proteins. So I'm wondering, did you check to see how many of those actually have strong co-variation signal between the chains? Like, did you
Yeah.
look at statistics like couplings? It's actually between pairs of residues across chains and said, oh, there is a really strong coupling here. But it's not
Yeah.
useful. Or, you know, worst case scenario is you picked all these examples that have zero coupling between the chains, and then of course it can't use it because there's no signal there.
Yes.
Period.
Yeah, so that's I guess what I'm trying to show a bit of on this plot on the left. So on the x-axis is that covariational signal with the pairing, and then on the y-axis is without that pairing. And so I guess it's, yeah, sorry, this is like a bit hard to intuit like what these values exactly mean, but even with like some of the structures where it has some of the most covariational signal, when paired after it's shuffled, it's still or sorry, it drops. Like things that sort of exist below this diagonal. So like this example here. Yeah.
Looks like a lot of your examples do have some covariation occurring between chains.
Right. Yeah.
Based on this mutual information.
Yes. Yes. Mutual information. But yeah, of course there are things that are like still on the diagonal, and I guess there we don't expect the prediction to change that much. Yes.
What if you try to put an incorrect peptide with no interaction? So you create an artificially thing that should not wrong. What kind of result are you getting?
Yeah, the question was what would happen if I used a peptide that isn't expected to work in this interaction, like an artificial peptide or some random peptide. I've done a bit of that in replacing the peptide with glycines and also shuffling the residues within the peptide. And I see something sort of similar to the mask test where especially with the peptide shuffled, it still puts it in like the same place. And very often so with the glycines as well. So yeah, it likes to put it in the same binding site, even with like I guess like a bad peptide. Yeah. But I don't know if I could say I know more than that for what happens. Yeah.
Does the PLDDT drop?
Good question. Yeah, the question is whether PLDT or PAE would drop with like the artificial or like the shuffled peptide. I actually don't know off the top of my head. Yeah. I saw it. Yeah.
So since the P specific attention to some of the NMR data, sometimes there are some dynamic information.
Yeah, the question was whether I looked at like NMR structures and the dynamics that you can find there. I didn't. Yeah, I kind of ignored NMR structures that didn't have just like a single reported structure. Like I didn't look at dynamics at all. Yeah. That would be interesting.
Like 3 questions online, but
Oh, yes. Okay, let's start from the beginning. Oh, yes. I'm sorry. The slides were clipped at the right, and yeah, I apologize. I don't know how to fix that. Have you looked at Dr. de novo targets? Can everyone see this question? Okay. Have you looked at peptides that dock to completely de novo targets and hence don't have an MSA? I think you briefly mentioned targets without MSA perform worse, but what about cases where the model can actually predict the target structure correctly? I have looked at, so in my set are a handful of structures where the target is a de novo design. I think there's a Baker lab structure on peptides binding to amyloidogenic targets. Anyway, yes, so in those cases, the target is predicted correctly and the peptide is predicted correctly, like it becomes a perfect prediction without the MSA. So those are the kind of special cases that we see with de novo. Yeah, with de novo targets. I can repeat that in the chat at some point.
Thank Lindsey again for great talk. Lindsey will stick around. You can pepper her with more questions. Feel free to grab the few drinks that are left. There's food going. Really cool talk. Really interesting.
Thanks.
What is a good? What is a sizable covariation signal in the product?
I hear.
I don't know what it. I don't know what 0.3.
No,
Yeah,
exactly.
yeah.
Yeah.
Go to one.
Yeah, is it?
No.
Is that like that seems
Does it go to
crazy.
100?
Yeah, old
Like
resolution.
what is it? So are they all like? Is it? Do you have some understanding that the like 0.3 or 0.2 ones are really strongly covariant?
Yes, yeah. So like from just looking at a bunch of these, I get a sense that that's true. I also applied some filters, like quality filters,
Yeah.
looking at like how many gaps are. So these aren't, will be like sort of substantial here MSAs. Yeah.
Okay.
But good question. I just presented some numbers that have no units and no reference. So yeah.
Because I mean, when you in that slide, you have like there's some small drop in
Yeah.
performance, and I'm just wondering if like the proteins that have the strongest coupling and the strongest drop, if you just took those proteins out, you no longer have a drop.
That's, I don't think that's, I don't see that. I have tried that. Yeah, I have an example here, actually. Sorry, I'm gonna stop sharing.